Hello students, today we are going to study a very important topic and that is known as the tRNA. Okay, and we'll be today studying about the tRNA as well as the structure of the tRNA. First of all, you must know that what is tRNA. tRNA means what? It is the transfer RNA. Okay, it is the transfer RNA. You must know that this transfer RNA is single stranded. Okay, it is single stranded. It has one end is having 5' end and another end is having what? That is known as the 3' end. It is single stranded, but due to too much folding, it seems to be double stranded. Double stranded like the DNA, but in actual it is not double stranded DNA. Due to folding, it seems to be double stranded because what happens when the tRNA is folded? So what happens? The nitrogen base pairs, complementary nitrogen base pairs come near to each other. They just come opposite to each other, and due to which it seems to be what? Double stranded, and actually it is what? Single stranded. The second thing is that tRNA molecule is the smallest, right? It is known as the smallest molecule. It is the smallest tRNA, or we can say it as that it is the smallest form of RNA. It is the smallest form of RNA, and it is made up of only 75 to 95 nucleotides. Okay, and that's why it is considered as what? The smallest form of the RNA, because it is made up of only 75 to 95 nucleotides. Okay, then 20 types of RNA present, tRNA present. Okay, 20 types of tRNA are found. Why 20 types of tRNA are found? 20 types of tRNA are found because 20 amino acids are found. 20 amino acids are there. So each amino acid is carried by each tRNA molecule. So altogether there are 20 types of the tRNA which will carry the 20 amino acids. Okay, now coming to the molecular weight. What is the molecular weight of the tRNA? So the molecular weight of the tRNA is 25,000 to 40,000 Daltons. Okay, its molecular weight is 25,000 to 40,000 Daltons. Okay, now it is also called as tRNA is also called as soluble RNA. Soluble RNA. Why it is called as soluble RNA? Because it is soluble in 1 molar NaCl solution. It is soluble in 1 molar solution of NaCl, and that's why it is known as what? The soluble RNA. Okay, it is also called as adapter. It is also called as adapter RNA. It is also called as adapter RNA. Why it is called as adapter RNA? Because it carries amino acid to site of protein synthesis. It carries amino acid to site of protein synthesis, and that's why it is also called as the adapter RNA. Okay, now next, the best model to explain structure of tRNA. The best model to explain structure of tRNA was given by, you must be knowing, Robert Halley. Was given by the best model to explain structure of tRNA was given by Robert Halley in the year 1965. Okay, and he gave a model, and that model is popularly called as clover leaf model. At present we are having the best model right, to explain the structure of the tRNA, and that is known as the clover leaf model. Okay, now you must also know that the tRNA also consists of some abnormal and modified bases. tRNA also consists of some abnormal and modified bases. I am writing few of here. The number one is known as pseudouracil. We will be discussing just now also. Na? So one is known as the pseudouracil. The second one is known as the inosine. These all are abnormal base pairs, inosine. And the third one is known as dihydrouridine. Right? Dihydrouridine. So pseudouracil, inosine and dihydrouridine, all these are what? Abnormal base pairs. And before starting the structure, you must know that transfer RNA is called as transfer RNA because it transfers the activated amino acid to the site of protein synthesis, and that's why it is called as what? The transfer RNA. Now let's have a look on the structure of the tRNA. That is, we are going to study the clover leaf model, which was given by Robert Holley. So I am writing here the clover leaf model. Now see here, if we study the clover leaf model, there are altogether 5 arms. Basically, there are how many arms? 5 arms. But out of the 5 arms, 4 arms are very important. Okay, so I am making the clover leaf model. See here, these are the arms of the tRNA molecule. Remember that tRNA is not double stranded. It is seeming to be double stranded, but it is not double stranded. It is not what? It is not double stranded. It seems to be what? It seems to be double stranded. Why it seems to be double stranded? Because of too much folding. Now this is the 5' end, this is the 3' end. These two end, 5' end and 3' end, suggest that it is single stranded. But what happens? Folding occurs. Say for if this is the tRNA molecule, so if it folds, so what happened? The complementary base come in front of each other and hydrogen bonds are formed like this type. Here hydrogen bonds are formed. Okay, now you are able to see here how many arms? 5 arms. 1, 2, 3, 4, right? And the 5 arms. This is the 5' end. At the 5' end, guanine is present. This question, from here the question is asked that which base is present at the 5' end of the tRNA molecule. So you will always say that at the 5' end guanine is present. At the 3' end, always remember, at the 3' end a special sequence is present known as CCA. At the 3' end, what is present? CCA sequence is present, and a functional group is present here. A free functional group is present here at the 3' end, and this free functional group is called as what? The OH group. So remember, at the 3' end a CCA sequence is present, and at the end of the 3' end a functional group is present known as the OH. Okay, now see, I will be talking about the arms here. I am talking about the first arm, right? First arm, this 3' end. This 3' end of the tRNA is called as activated. It is called as activated amino acid attachment. It is called as activated amino acid attachment site. Means the 3' site, the 3' end is the end of the tRNA where the activated amino acid will come and attach. That's why it is called as activated amino acid attachment site. So that's why the first arm is very, very important. Okay, now coming to the second arm. Now the second arm is having approximately 7 bases. It is having how many bases? 7 bases. and this arm is having, this is called as what? The stem. This is called as from here to here, stem. And this is what? The loop. Always remember, the stem is the part where the hydrogen bonding occur, and loop is the part where hydrogen bonding does not occur. Hydrogen bonding does not occur, doesn't occur, right? The place where the hydrogen bond is not formed, at that place loop is formed, and in the stem part what is formed? Hydrogen bonds are formed. Okay, now this arm is also called T psi C arm, or you can call it as T psi C loop. This is called as what? T psi C loop. Now let's have a look. What do you mean by T? T means thymine. Now always remember, RNA is not having the thymine, it is having the uracil. In place of thymine, what is present? Uracil is present. But here in this arm, particularly here in this arm, thymine is present. Now what is this sign? It is called as what? Pseudouracil. It is called pseudouracil, right? And this pseudouracil is what? So just now I have talked about abnormal base pair. It is a abnormal base pair. Okay, it is a abnormal base pair. Okay, or we can say that abnormal base which is formed here, and this abnormal base is responsible for the formation of the loop. Okay, you know very well the loop is the place where the hydrogen bonds are absent. Now the question arises, why at this region the hydrogen bond has not formed? So hydrogen bonds are always formed between the complementary bases, but this is an abnormal base. Pseudouracil is an abnormal base, and that's why at this arm what is formed? A loop is formed, right? So T means thymine, this one is pseudouracil, and C means what? The cytosine. All these are found one after another. Okay, now the question arises that what is the function? What is the function of this arm? So the function of this arm is that with the help of this arm, tRNA combine with larger subunit of ribosome, You know how many units of ribosomes are there? 2 subunits. Say for this is a small subunit of ribosome, and this is a larger subunit of ribosome. This is larger subunit of ribosome. Okay, so always remember, this tRNA molecule combined with the larger subunit of the ribosome with the help of this arm. It combines with the larger subunit of the ribosome. This is what? Larger subunit of the ribosome. So what is the prime feature of the T psi C loop? The prime feature of this arm is to just attach the tRNA to the larger subunit of the ribosome. Okay, and this arm consists of what? 7 bases. Now this arm is called as what? Variable. This is what? Variable arm. It is a sort of lump you can see here. Okay, it is a variable arm and not having so much of function. It is a variable arm. Okay, now this is what? Now I am putting the numbering here. This is the second arm. This is the second arm, T psi C arm. Now coming to the third, which is the most important arm of the tRNA, and that is known as what? Anticodon arm oblique loop. Okay, why anticodon arm? Because this arm consists of anticodon. Now let's have a discussion on the anticodon. Just think of that anticodon recognize codon on mRNA. Anticodon recognize codon on mRNA, right? And what is the type of the anticodon? According to that anticodon only the amino acid will come at the 3' end. Okay, so the anticodon decides the type of amino acid which will attach to 3' end, Why? How it recognizes? To try to understand this, say for this is a mRNA, which is having one end 5' and another end 3'. Now think of that on mRNA, you must know that on mRNA what are present? Codons are present. Now see here, say for this is AUG. mRNA is having what codon? This is what a codon. If you have studied the genetic code in the class 12, you might be knowing that a codon is a three-letter language. A codon is a three-letter language made of nitrogen base, which is having the genetic information to code a particular amino acid. Okay, so if see here, this is the codon and the anticodon is always present on the tRNA. Okay, and codon is having the complementary sequence that of the anticodon. Say for if A is here, then instead of A, what will be there? U, because we are studying what? RNA, and in RNA thymine is not present. That's why there is uracil present. So U, so A, G, and then C. So this is what? This is a anticodon. So anticodon present on the tRNA recognize the codon on the mRNA, and due to which what will happen? The type of the anticodon will decide that what type of the amino acid will come and attach at the 3' end of the tRNA. Why? Because anticodon recognize codon, and codon is having the genetic information for a particular amino acid. So that's why this arm is very important. But at this arm also you can see that here at the stem region, you can see that hydrogen bonding is occurring. But what about here? Here what is formed? Loop is formed. Now the question again arises in mind that why the loop is formed here. You know very well, right, that just now I have said that loop is the place where the hydrogen bonding is not there, and hydrogen bond is not formed because of the abnormal base pair. Now the question arises, which abnormal base is present at this arm? So you must know the abnormal base which is present at this arm is called as inosine. Is called as what? Inosine. Okay, inosine. I am writing here, it is what? Abnormal base. It is what? The abnormal base. So loop is formed because of the abnormal base, and I will be putting here the number 3, right? So this was the first arm, this was the second arm, this was the third arm, known as the anticodon arm. Okay, now coming to the last arm, and that is known as, this is known as D loop. This is called as D loop or arm. This is called as D loop or arm. Why it is called as D loop? It is called as D loop because it consists of dihydro. It consists of dihydrouridine. It consists of dihydrouridine. Right? Now the question arises, what do you mean by dihydrouridine? In short, sometimes it is also called as DHU, and that's why instead of D loop, sometimes we call it as DHU arm. Why it is called as DHU arm or D loop? Because it consists of dihydrouridine. Again, this dihydrouridine, you know that it is an abnormal base. It is an abnormal base, and always remember, whenever the abnormal base comes, then a loop is formed and hydrogen bond is not formed here. So here there is abnormal base. Once again I am repeating, here the abnormal base pair was the pseudouracil, here the abnormal base pair was the inosine, and here the abnormal base is known as what? The dihydrouridine. Now this arm is called as the fourth arm. Okay, this arm is called as what? Fourth arm. We can also call it as, this arm is also called as amino acyl synthetase. Amino acyl synthetase binding site. This is called as what? amino acid synthetase binding site. This site is also called as amino acid synthetase binding site. Now the question arises that what is amino acid synthetase? So amino acid synthetase binds here. Now amino acid synthetase is a enzyme. This enzyme recognizes and this enzyme recognizes and activates amino acid. This enzyme activates and recognizes amino acid. This enzyme recognizes and activates amino acid so that amino acid is attached to the 3' end of tRNA. So this is also very important. Amino acid synthetase binding site means at this site amino acid synthetase enzymes bind. This enzyme is very important for the recognition of the amino acid, as well as it is important for the activation of the amino acid. And as soon as the amino acid is recognized and it is activated, the amino acid will come and attach at the 3' end of the tRNA. And this arm is also called as D loop because it consists of what? Dihydrounity loop. So this was the famous cloverleaf model for the tRNA, and it was given by Robert Holley in the year 1965. Okay, and before ending the tRNA molecule, you must know that it is called as the transfer RNA because it transfers the activated amino acid to the site of the protein synthesis. Okay, so keep watching my videos. We'll be coming with new videos. In the next videos, we will be dealing with the other types of the RNAs also, such as the ribosomal RNA and the messenger RNA. Thanks a lot.